right, well, good afternoon, everyone, and welcome to today's EOL seminar. I'm Sarah Woods, and on behalf of EOL, I'm pleased to introduce Dr. Russell Perkins. Dr. Perkins received his bachelor's in biochemistry before obtaining his doctorate in physical chemistry with a certificate in molecular biophysics from the University of Colorado at Boulder. Currently, Dr. Perkins works as a research scientist at Colorado State University, where he leads complex and collaborative ground and aircraft-based aerosol and air quality field experiment campaign measurement efforts. Dr. Perkins has been recognized for his teaching with multiple physical chemistry graduate teaching excellence awards and was more recently recognized for his research efforts with the 2022 CSU Walter Scott Junior College of Engineering Rising Star Researcher Award. Russell and his team have been kept quite busy collecting both ground and airborne measurements of ice nucleating particles in recent years, which EOL might recognize through NSF's Spicule and Caesar, DOE's SAIL, as well as NASA's ARCSIX campaigns. And despite his busy field schedule, a fun fact, somehow through it all, Russell has found the time to become a licensed drone pilot. Please join me in welcoming Dr. Russell Perkins for his seminar titled Key Findings from Recent Field Campaigns Examining Environmental Ice Nucleating Particles.
Thank you so much for the invitation and the lovely introduction. So I'll go ahead and get started. Let's see. All right, so I'm going to start with some motivation. We're talking about ice nucleating particles today, and the motivation for that really comes from the fact that cloud phase and droplet concentrations are important for a lot of different processes like precipitation and cloud lifetime and radiative energy balance. The phase and concentration of clouds is controlled at least partially by aerosols. And where that control comes in is a couple of different places. So first, cloud droplets only form on existing aerosol particles. And in a kind of analogous process, pure liquid droplets won't freeze until temperatures that are quite cold, around minus 38 Celsius, without an existing aerosol particle of a special class called an ice nucleating particle, or existing ice. We'll talk about that a little bit later. And the reason for both of these things is actually very similar. So if you think about forming a droplet, when you get to 100% relative humidity, then liquid water becomes the favored phase over gas phase water. And similarly, when you get to 0C, ice becomes favored over liquid water. And that's true in kind of the bulk of the center of a droplet. But you have issues making an interface. So right at the edge of the droplet, you have to pay essentially an energetic cost to form an air-water interface or to form a liquid-water-ice interface. And that cost is really the reason that these processes don't happen spontaneously as soon as it's thermodynamically favorable for the bulk. And so you have aerosol particles essentially reducing that cost and allowing these phase changes to happen. And so with ice nucleating particles freezing of cloud droplets, there's a couple of different kind of categories that I'm going to talk about. The first is primary ice production. And so primary ice production is freezing of a liquid droplet that's initiated by an ice nucleating particle, by one of these aerosols of a special class. And there are lots of different ways that freezing can happen. We're mostly going to stick to this mixed phase regime in between 0 and minus 38 C. And the reason that that's mixed phase, right? Because it's possible both for supercooled liquid and for ice to coexist. At warmer temperatures, you don't have ice, and at colder temperatures, you can't really have supercooled liquid because it'll freeze spontaneously. And so kind of the classic way that you get freezing by an ice nucleating particle in this regime is that you have activation as a cloud condensation nuclei to make a cloud droplet. That cloud droplet cools and eventually reaches a temperature where the ice nucleating particle can activate, and you get freezing of that droplet. And that's essentially what's categorized as immersion freezing. And there are a few different kind of variations on that. There's condensation freezing, which happens when you condense liquid and you're below 0 C, but it's often lumped together with immersion freezing because the two processes are not greatly different in many situations. And the reason that we look at immersion freezing is that we have some evidence that it's the most important freezing mechanism for producing primary ice in mixed phase clouds, or in this mixed phase temperature regime. And yeah, in this, just to reiterate, kind of in this temperature regime, all primary ice is formed on an ice nucleating particle. And then I think it's important to kind of stop and think a little bit about what we actually need to know about primary ice production and how well we need to know it. And some of these are hard questions, but I think generally we need to know this temperature-dependent INP concentration curve. So different INPs will activate at different temperatures, and we'll see a lot more of that. And it turns out that these things are fairly hard to measure, and so I think there's some open questions about how well do we need to measure these concentrations. Right now, measurements that are good in INP space are within a factor of 2 uncertainty, and a lot of the time it's an order of magnitude or worse, just because these things are fairly hard to measure, and we'll talk more about that later. I talked already about measuring immersion freezing because we think it's the most important, but I think there's open questions about if this is good enough in order to represent these processes in models accurately. And part of the reason is that, well, there's just a lot of uncertainty in figuring out if we're doing a good enough job. But then beyond getting those measurements at one point in space and time, we need to understand at least some things about the spatial and the temporal variability of ice nucleating particles. And so I think about this kind of on the coarse and the fine scales. So, coarse spatial variability is going to be regional differences, long-range transport, whereas fine scale might be gradients around clouds, things like that. And then temporal variability, similar considerations. We need to get seasonal and diurnal cycles for representing these particles and primary ice production. But then we also have some evidence that fine scale processes, how they're transported within clouds, how they're depleted out of clouds, can be very important. And then the last thing on here is maybe one of the trickier things is there's potentially actually time dependence for activation of a single aerosol particle. If you have that in liquid in a droplet of a certain temperature, it might take some amount of time to freeze. And we're not going to talk about that today because it's hard. And a little beside the point for kind of current modeling efforts, but. All right, and then to make things worse, this is part of the reason that it's hard to figure out how well we need to understand primary production is because it gets really convoluted because you can have secondary ice production through a lot of different mechanisms kind of shown here. And so the idea, the difference between secondary and primary ice production, you have an existing ice particle that's causing a new droplet to freeze. And so, maybe the most classic example is the Hallett-Mossop process, where you have rime splintering. You have small shards of ice that are produced from this riming particle, and they can go on and impact other liquid droplets and cause them to freeze, because ice is the best ice nucleating particle. And so this gets complicated because there are a lot of different secondary ice production processes. And many of them are not well understood or well parameterized. Like some of these are kind of more in the realm of thought experiments than anything else. And I think it's very likely that there are secondary ice production processes that happen that we are missing entirely. And so this makes the connection between primary ice production and ice content of clouds, cloud phase, a little trickier to figure out. But one of the reasons that we still care about primary ice production as much as we do is that we know that all of these secondary processes are going to depend on primary ice production happening. You need to have some ice before any of the secondary production kicks off. And then another thing that's particularly interesting about this is that the secondary ice processes may be much more sensitive to the ice nucleating particles that are active at the warmest temperatures rather than necessarily the temperature of the cloud. So that's mostly what I'm going to say about secondary ice production, but I think it's important for understanding, for kind of interpreting our results and for passing along any of our findings into modeling work. All right, and now we're going to take a break and play a fun game called Find the INP. So we've got 1 million dots on this screen, and all of the blue ones are normal aerosol particles, and the red ones are ice nucleating particles that are active at about minus 20 C. And there's one of them down there. I promise they're not all blue dots. And so the reason I like to show this is because I think it's a really good example of how ice nucleating particles might not strongly correlate with bulk aerosol properties. And this is something that's really hard to deal with because you could see your aerosol composition change dramatically and maybe your ice nucleating particle concentrations stay exactly the same because there's some subpopulation that really is not related to the bulk aerosol. And then the other thing that this means is that measurement statistics are very difficult. And we'll talk a little bit about that next. So despite these things being rare, we do have some good measurement techniques for finding them. And the first one we call the ice spectrometer. It's an offline INP measurement technique. And so offline in that we are collecting aerosol samples on filters, generally, pulling air through a filter. Here's a picture of our setup on the top of an aircraft rack for airborne field campaigns. And then we'll take those filter samples, resuspend the particles in liquid. And then the idea is we have some options for what we do next. In the most basic version, we take that resuspended particle sample in liquid, we aliquot it out into repeated aliquots, and then cool those down and watch as the different aliquots freeze. We can take those frozen fractions and ultimately back out an ice nucleating particle concentration per volume of air that was sucked through our filter sample. And then we have a couple of other options to get a little more information about the types of INPs that we're sampling. The first is heat treatment, and we do this because it will selectively denature and destroy biological ice nucleating particles. Things like proteins will change their folding and they'll no longer be active. And then we have another treatment that's even more aggressive to essentially mix in hydrogen peroxide and chew up all of the organic material. And so by looking at how our INP concentration changes as a function of the treatment, well, I should take a second to explain these graphs. So we have temperature on the x-axis. So when you move to colder temperatures, you should always see more INPs active at colder temperatures, with the idea being this is essentially a cumulative spectrum. So if you have an INP that freezes at minus 10, it will also freeze at minus 15 or colder. So the treatments will effectively destroy all of the INPs of a certain class, and then you can look at the difference between the untreated and the treated version to look at how many INPs were in that that class of materials. So we get our heat label organic INPs, which we often assume to be biological in nature, and then these heat stable organic INPs, which are probably ultimately biological as well, but they may be older, more degraded, things like that. All right, and then Our more real-time technique is the continuous flow diffusion chamber, or CFDC, and it measures INPs in real time and in a way that is pretty analogous to how we expect freezing to happen through the immersion mode in clouds. This one, we use an upstream impactor to take out large particles because we're ultimately trying to distinguish INPs from normal aerosol particles by their size at the end. So the smaller aerosol particles get passed into this chamber where we have iced walls at different temperatures in order to achieve a water supersaturation at temperatures below freezing. And so we get nucleation of all of the aerosol particles into droplets because we're significantly supersaturated. And then those that contain ice nucleating particles active at our measurement temperatures will freeze. They'll start making larger ice particles. And then we have this droplet evaporation region where we're still below 0 C and above ice saturation humidity, but below water saturation. So the liquid droplets evaporate. And anything that's large that gets sized at the end, we can be fairly confident is an ice nucleating particle. All right. And then we have a couple different sampling strategies that we've employed for different campaigns. The simplest is this kind of ambient sampling. I have some examples of inlets for the C-130 that were used during the Caesar campaign. So this ambient or isokinetic sampling is what we use for our offline samples. We collect material onto filters. And then we can use that directly with the CFDC, or we can use an aerosol concentrator as a trick in order to improve our sampling statistics. And so that's kind of a neat thing. It's like a giant virtual impactor. And the idea is that it takes in a huge volume of air, 250 liters a minute, and it tries to concentrate all of the aerosol in that air into a small flow that then goes to our instrument. And so we often get concentration ratios on the order of 50 or more for large aerosol particles above about 500 nanometers. And so this is nice because it ultimately improves our detection limit with our CFDC, and we can also use it to improve our temporal resolution if we have enough material. And then the last thing that we can do is sample downstream of a CVI on an aircraft in order to take in cloud droplets and look at the aerosol content of those droplets rather than the kind of interstitial air. All right, so that's what we're measuring and how we're measuring it. And now I'm going to talk about a couple of different campaigns. The first one is this Surface Atmosphere Integrated Field Laboratory, or SAIL. This is a Department of Energy sponsored project through atmospheric radiation measurement and atmospheric systems research. There have been a lot of people involved. I have just the people leading the kind of main campaign. There are lots of projects associated with it. There have been a couple of co-located campaigns through NOAA and NSF, the SPLASH and SOS campaigns. And this project really had an overall kind of hydrology and hydrometeorology focus. And the place that we fit in is that the INPs and the cloud ice ultimately impacts precipitation. And so, yeah, for our end of the project, I've highlighted the people in our group that did a lot of the sample analysis and a lot of the data analysis, especially Richen Zhao. So this campaign happened near Crested Butte, Colorado, from September 21 to June 23, so it's almost 2 years. And there's a lot of different samples that we took advantage of. There's samples collected through ARM, and some of these were analyzed, and some of them we got and worked with to perform additional analysis. And then there's this distributed network site that Handic Scientific set up called SailNet. And so all of those purple hexagons are different SailNet sites with some instrumentation with the idea being that we can look at kind of spatial distributions of INPs and aerosol. And so they're max 16 kilometers and 570 meters in the horizontal and vertical apart. And the other thing that's, I think, really interesting, well, there's a couple things that are really nice about this site. One is that there's a lot of regional cloud seeding, and this is very common in, I guess, all of the western states at this point. So all of these blue triangles are seeding sites where they're intentionally trying to produce ice nucleating particles for the purpose of glaciating supercooled liquid clouds and increasing precipitation. So we expect to see some anthropogenic INPs here, unlike a lot of parts of the world. And then there's an improved network site that's relatively close, and we took advantage of that to do some source apportionment for this region. And so, it has a nice long-term data record. It has a lot of aerosol composition. And so with PMF, we can pull out a few different aerosol types that we see. So we see sulfate and nitrate, presumably secondary aerosol, but then we also get a lot of biomass burning and dust. So these are the main sources in this region. The sulfate and nitrate look pretty typical for the sale period. Biomass burning is lower than normal for this time period that we're looking at, so that's important to keep in mind. And then the dust contribution is maybe a little bit higher than normal, although both of these things tend to be pretty episodic. All right, and here's a terrible plot just to show you that I have lots of data. So rather than that INP concentration versus temperature, now we have that split out into different traces so that we can have a time axis on here. This is mostly just to show that it's a relatively nice long-term data record. There's, these are all from offline samples, and there's definitely some spacing in between samples, but there's enough to get at kind of the seasonal cycle at this site. And if we kind of take that data and compress it down into a monthly average INP concentration, we get some strange results. And most notably that we have our highest INP concentrations in like December. We get big increases in November in some years. And so this is a little puzzling if you didn't pay attention to the seeding activities that are happening here. So this is an INP spectrum from what we have marked as a cloud seeding day through a combination of how the spectrum looks, and then also we have generator logs for the sites that are nearby. So we see very consistent spectra for the days that are logged as days where the generators were running for long periods of time. There's a couple puzzles in here, and one of them is that we're getting our freezing onset at colder temperatures than we expected. Silver iodide generated in kind of a laboratory setting will freeze around -4, -5, and then it plateaus. And so, there's maybe a question about whether this sort of hump that we see in there is due to that silver iodide. I told you before that heat removed biological materials, and we see an impact here. Silver iodide's kind of tough because a lot of the particles are really small, on the order of 50 nanometers, and it is a slightly soluble salt. So it's at least possible that we're essentially just dissolving the silver iodide particles with our heat treatments. But kind of beyond that, there have been some studies that have found similar sort of activation characteristics for silver iodide particles that we're seeing here. And then there are a lot of things that can really impact the way that these particles are generated and their efficiency as ice nucleating particles, from the solutions that are burned in the generators, the impact of wind while the solutions are being burned. And then, because we're not measuring right downstream of a generator, this is kind of maybe a more regional concentration. I think there's there's definitely an open question about whether we're seeing depletion of the most active silver iodide INPs because they've all frozen in supercooled liquid clouds and precipitated out. So it's not totally unambiguous, but I'm at least fairly confident that what we're seeing is at least a seeding signature. And then we see another interesting signature that doesn't look like silver iodide that shows up in the wintertime as well. And this looks kind of like a classic biological particle. Essentially, when we have this really sharp activation, that means we have a bunch of things that are active. They kick in at the exact same temperature range, which is very characteristic of biological materials. In particular, there is a bacteria called Pseudomonas syringae, which is a well-known ice-nucleating bacteria, and it actually gets used commercially in snowmaking as a product called SnowMax. So it's dead lyophilized Pseudomonas syringae, and it activates essentially right at the right temperature. It shows what we expect for a heat treatment sort of decrease. And so we're fairly confident that at least on some of these days, our INPs are actually dominated by INPs released from snowmaking activities at the nearby ski resort. And so if we take those things out, those days that we've flagged either as snowmaking or as cloud seeding activities, we get a seasonal cycle that's much better behaved. And so I think there's an important thing here to keep in mind that the actual seasonal cycle looks like what I showed before. But if you wanted the kind of natural background INPs without those activities, you get something more like this, where you get kind of a peak across all of the temperatures in the summer, and there is some variability in here year to year, and then you tend to get your lowest concentrations in the winter. So we did all that source apportionment earlier, and so if we compare kind of the different sources from the PMF with what we're getting for ice-nucleating particle concentrations, we get some broadly consistent data. So, none of these traces match up perfectly, but we at least see the same, basic characteristics in the aerosols we were seeing with the INPs. We see higher concentrations of dust and biomass burning in the summer and lower concentrations in the winter. And so I already told you that INPs don't necessarily track with bulk aerosol characteristics, and then I'm going back and doing just that. And so I think the thing to remember here is that we don't necessarily expect these correlations to be perfect, but if the materials are broadly coming from these sources, it makes sense that we see some of these relatively good correlations. And then the other thing that we can look at, so it does look like maybe they're related to dust and biomass burning, but then if you look at our sort of treatment data, at minus 15C, we have a lot of things that show up in this kind of biological material category, and it stays pretty prevalent across our entire temperature range. We see more of the organic stuff growing in at colder temperatures, but really the inorganic component stays pretty small. And so there's something interesting here where even though dust might be kind of what we call the source, maybe the mineral type materials are a large component of the aerosol mass, for arable soils and things like that, you often get biological materials associated with the dust as it's associated with and like co-emitted internally mixed with the dust. And I think that that's probably what we're seeing here. And so we've seen this for some previous campaigns. The one on the right is from the cacti campaign in Argentina, where there was a lot of arable soil dust. And you see a fairly strong correlation in between the inorganic ice-nucleating particles, so the dust, the mineral dust type component of the aerosol, and then the heat stable sort of organic components, kind of backing up this picture. This one on the left is for the sail data. And rather than a 1-to-1 line, we have a 10-to-1 line. So there's an even, I mean, the correlation is maybe similar, but there's relatively more organic material in what we're getting at the sail site in the mountains than what we were getting in the cacti site. So that's kind of interesting. It's not a tremendous difference, but it is significant. You know, we're generally seeing more organics and more biologicals at this mountain site. And then we have a little bit of single particle composition. So this is an effort led by Swaroop Jina at PNNL. And this is computer-controlled scanning electron microscopy and elemental composition analysis from that. And so they have some algorithms to kind of classify individual particles in their microscopy to different types. And we do see, you know, essentially a lot of carbonaceous aerosol in some of these different samples. And, you know, so in the first February sample, there is this large carbonaceous peak. There's not much straight mineral dust, but it does look kind of like an arable soil. When we get to this June sample, there's more of that dust growing in. We get a lot of sulfates kind of all of the time. And then we have some of this sodium-rich material that's kind of interesting. We're not quite sure what that is at this point. One option is like playa dusts. So you can get these sodium-rich kind of mineral dusts. But then another that's particularly interesting is sometimes biological particles can actually contain a large amount of sodium. But they've just updated their kind of machine learning classification scheme to include more biological components. And so some of this data might change as that gets run as well. All right, so we have a lot of data. We have some ideas of what our source materials are, and now we kind of want to try to tie it to something. And so one relatively standard practice these days is converting from an INP concentration and just an amount of INPs per volume of air into an ice-active surface site density, which sounds really complicated, but the idea is just that you're kind of normalizing your INP spectra by your total aerosol surface area. And at least for this campaign, it works quite well, I think. So you can see the spread of our different, well, okay, so I guess the first thing is all of the purple traces are cloud seeding spectra, and all of the blue traces are days where we have influence by snowmax. And those kind of diverge from everything else when we do this normalization, and that makes sense because those are like intentionally produced efficient ice nucleating materials, so we would expect them to have higher ion active surface site densities. Looking just at everything else that's left for our kind of natural samples, the spread tends to be reduced. And there are a couple of outliers, but you know, we get to a point where we have maybe an order of magnitude spread between our different samples, whereas we had maybe 2 orders of magnitude just in concentration space. And so this is nice kind of for 2 reasons. One is that we maybe have learned something about how to track our INPs, and the other is it gives us a good way to kind of parameterize them to tie them to something that's easier to input into a model. And so there are quite a few other parameterizations in this space to look at how you can tie the aerosol surface area into ice nucleating particles. For this campaign, we get relatively lower surface site densities than for other parameterizations of things like agricultural soil and dust, but it's still significantly higher than existing parameterizations for biomass burning aerosols, which is kind of what we expected. Biomass burning aerosols tend to be relatively inefficient INP sources. And yeah, there is relatively less spread than what's often been seen and used to build some of these other parameterizations. It's fairly well behaved. We have a very similar shape, it just is slightly less efficient. And that might be a hint that what we're seeing is INPs that are potentially dominated by long-range transported sources rather than local ones, at least for cases outside of the cloud seeding and snowmaking. And so we have that distribution of sites. So let's look at some of what we're seeing there. And at least kind of on the scale of INP measurements, a lot of these spectra look pretty similar. There are a couple of things that tend to fall out. So this snodgrass site tends to show up at lower concentrations than everything else. And this one's kind of funny because snodgrass is the site that's right in the middle of everything else, and it's also in the middle of elevation as well. And so the reason that it ends up being the lowest concentration is kind of puzzling. We have some ideas maybe related to the flows that happen around the mountains there. But I think overall, there's less spatial heterogeneity than we expected. And we've actually seen some similar things. So in the DOE tracer campaign, we had a couple of sampling sites that were, fairly far apart, 76 kilometers. One of them is, essentially in an urban coastal environment in Houston, and the other is in this agricultural environment outside of Houston. And so we really expected these things to look pretty different. If you look at the INP spectra, you see that they're very strongly correlated with the two different traces in each color, kind of across the whole temperature range. And so at least for that site, I think it really suggests that we have these situations where ice nucleating particles are really regional in nature. They have distant or uniformly distributed sources. And so that's the same sort of picture at the sail site right now. We have more analysis to do to kind of dig into that a little bit more, but it is kind of interesting. And so to summarize this part, we got a nice annual cycle. We saw, using kind of the treatments and the aerosol observations, that things were relatively well behaved, right? We saw things like dust as a major source. We saw relatively little spread, very consistent freezing spectra, shapes. We maybe saw some biological influence, but it was relatively well distributed. We saw big influence of cloud seeding and snowmaking activities, that were very episodic but very important for understanding populations of these things in the winter. And then we ultimately ended up with a situation that was fairly easy to simplify as far as building a parameterization that can be used for a model, in a very similar vein to what has been done in the past. And so sometimes it's nice when things work out without having to make them so much more complicated. And so kind of getting back at this picture of what we really need to know, I think we made some progress in these temperature-dependent INP concentrations from these measurements, but then we also got at kind of the coarse scale variability, I would say, with regional differences and seasonal cycles, with these measurement points that were, sometimes a week or two apart. So, and this coarse scale variability, in this relatively simplified view is pretty well captured by the bulk aerosol distributions and evolutions, which is kind of a nice, clean result. But things aren't always quite that simple. Let's see. Maybe I should think about how much time I have. Yeah, so we'll go through this part fairly quick. If you think about a thunderstorm out on the plains, how is our aerosol population that we've been measuring with maybe our long-term measurements going to change as that thunderstorm moves over us? And how are things like fluxes going to change? Why? And what measurements are really going to be the most relevant? So are the kind of long-term averaged measurements going to be relevant for the aerosols that make it into this thunderstorm? And so that was kind of the idea behind this BioAerosols and Convective Storms campaign that was collaborative between CSU and University of Iowa. Sean Freeman's at Huntsville now. He was at CSU at the time. And there's a lot of interesting things in here. We've talked about bioaerosols a bit, so I'm going to skip over that. Oh, actually, one of the interesting things here is that pollen can be a particularly good ice nucleator, and then if you get it wet, it can rupture and release all of these subpollen particles, which retain their ice nucleating ability. So it was one that was of particular interest going into this campaign. And so one of the kind of dynamic aspects that we were targeting is cold pool generation. So the idea being once you have rainfall, some of that rain evaporates and produces this cold, negatively buoyant mass of air that moves along the ground. And there's been previous work looking at how this kind of gust front can loft aerosol, in particular things like dust. And so we were really interested in this same sort of setup, but for biological aerosols. And so we had a lot of questions going into this about, do you get bioaerosol release in front of the storm? Do you get pollen and plant matter and bacteria? Do these things get ingested into the parent storm? Do you get pollen rupture, which greatly increases the impact of pollen? And then do you get release after the storm? There's evidence that you get more fungal spore release during wet conditions. And so there's a lot of different things that can happen here. This campaign took place in northern Colorado at the Central Plains Experimental Range. It's a USDA site. We partnered with NEON to do some measurements at their tower. And it's primarily grassland, grazed by cattle. It's fairly representative of the Colorado Plains. I'll skip over some of these measurements. And so just trying to look at ice nucleating particles at some finer temporal scales. This is now with continuous flow diffusion chamber measurements. This is concentrations at, I think, minus 20 C averaged every minute. And so there's a couple things that happen here. First, we have a cold pool come through in this first kind of purple box. We see an increase in wind speed, a rapid decrease in temperature, kind of characteristic of cold pool passage. And we see a relatively large increase in INP concentration on the order of a factor of 20 or more. And at the same time, we see an increase in fluorescent particles, which are used as a marker for biological particles, both in kind of the absolute concentration and then as the fraction. After that cold pool passes, we get rainfall, indicated in these light blue bars for rainfall amount. And as that rainfall happens, you get a huge increase in ice nucleating particles. You're more than a factor of 100 above the ambient concentration. You get a similar increase in biological aerosol, but not necessarily in the fraction. And then we have another cold pool passage that happens at the end, where ultimately by the end of that cold pool passage, our ice nucleating particles have dropped back down, not quite back to baseline. And so there's a big question of, if these convective storms are generating all of these ice nucleating particles, either with their cold pools or with rainfall, do these things ever get lofted to a point where they can cause freezing? For the rainfall-generated INPs, these are likely due to physical droplet impaction with the ground lofting material. And we see this in the aerosol measurements too. It's kind of interesting. It starts raining and your coarse mode aerosol goes way up. So like you might expect wet deposition to be cleaning the air, but you see the opposite, at least on the ground. We can look at the different ice nucleating particles before and after rain, and this is a 24-hour average sample split before and after. And we see this huge increase in ice nucleating particles, especially at the warmer temperatures. It's this rapid increase, and then it's relatively flat. We have heat treatment data for this. It all goes away with heat treatment, indicating that this is all biological ice nucleating particles that are being released with rainfall, or at least in this period post-rain. So a lot of this could be fungal spores that come out once the ground is wet. We have size resolution for these samples. One thing that's kind of interesting, so before rain, we have these different size bins, so 0.25 to 1 micron, 1 to 2.5, 2.5 to 10, 10 to 25, and larger than 25. And it's actually kind of interesting that the relative importance of all of these very different aerosol size categories is kind of similar, that they're as close to each other as they are. And we see in the post-rain sample that some of these bins are selectively enhanced. So essentially the three smallest bins between 0.25 and 10 microns. So 10 microns would be like an intact fungal spore. 0.25 to 1 micron is almost certainly a fragment of something, either a fragment of a bacteria or a fungal spore that's retained its ice nucleating ability, or plant matter, things like that. So this is super interesting. We actually have chemical tracer measurements for these same samples, and on a lot of these you see increased mannitol, which is a good marker for fungal spores. Less of the markers for pollen than we expected. So a lot of evidence that some of these are fungal in nature, maybe soil fungus, things like that. All right, and then kind of getting at the question of, can any of this INP-rich air make it into the parent storms? We have some simulations done by Charles Davis and Sue Vandenhever's group using the RAMS model. And so we're gonna have a storm moving across. This one is a supercell, and we're kind of looking at trajectories of parcels moving into the storm. So what percentage of parcels from a given area make it into the storm? And we've split this up between not rainy parcels and rainy parcels. And here, I'll just show all of these at once. So you essentially get these parcels of air generated out in front of the storm. This is relative to the storm center. And in the not rainy case, you get a lot of these parcels out in front of the storm that are getting entrained into higher altitudes where you get cloud formation and precipitation and all that good stuff. And for the rainy case, the regions where you get active rainfall, that you get these parcels, you actually do get a significant number of them from a certain part of the storm that get entrained. And so, there is a lot of variability in this. Some regions are gonna get entrained more than others. It probably depends on the specific storm. But there is something very interesting here where, at least in some cases, you might be able to entrain those INPs that your storm is generating into that storm. Let's see. We have similar things for a squall line case, and you get a very similar sort of entrainment, where it is at least possible that as you're generating these materials, they're getting entrained. And let's see, I'm pretty much out of time, so let's go ahead and skip past some things. Okay, so we see pretty good evidence for a lot of these processes that we set out to investigate. We definitely see some pre-storm bioaerosol release. We see changes with cold pools. We see some evidence that you can ingest these materials. We don't see so much evidence for pollen rupture because pollen ended up being a less important player than we expected. And we definitely see a lot of evidence for post-storm bioaerosol release, which also has implications for subsequent storms, maybe on that day or on later days. And one of the things that's interesting, I didn't show enough cases to really get this point across, but there's a lot of variability in how the INPs respond to these sorts of systems. So sometimes we'll get cold pools come through and the INPs go way down, or sometimes they stay the same. And so, with kind of what we've figured out at this point, oh yeah, and the rainfall ends up being very important as well. It's hard to kind of, we need to do a lot more work before we can try to put any of this into larger scale models. But we have learned some information about kind of the fine scale variability in INPs from this campaign. Yeah, but clearly more work needs to be done. All right, and I thought I had another section here. Well, maybe we'll just leave it here and take questions.
Thanks, Russell. That's a lot to cover, and it looks like you've got a lot more data to get through.
Yeah, I made too many slides.
We've all done that. Quick reminder to those online, there's a Slido box with a tab at the top of your video screen. You can enter your questions in there. Do we have any questions in the room?
Thanks so much for that presentation. So I was wondering on slide 38, you referred to the fraction, and so so this NF over NT, and it stays relatively constant while the INP concentrations are spiking. So what was that fraction?
Yeah, so that fraction is a fraction of fluorescent particles. So it's a number of fluorescent particles divided by a total number of particles. And in this case, it's greater than 500 nanometers, so coarse-ish mode. Yeah, so I think that's essentially showing that the composition of the aerosol is staying pretty similar. You're just getting a lot more of it.
Okay.
And you see kind of a similar thing. You can almost see it just if you look at the average fluorescent particles in black here versus the INP concentration. They're fairly well correlated for as they go up and down, up until you get towards that point there. But yeah, so I think it's showing maybe that it was the same sorts of ice nucleating materials that we saw come in with that first cold pool as we were getting during rainfall, which probably means that the INPs in that cold pool were from rainfall somewhere else, right? Maybe further upstream in this same storm.
Okay, yeah, cool. And then I was also curious about how the sizing works for this data that you collected.
Yeah, so this data isn't sized. This is separate offline data. I didn't go through any of the explanation of that. So this is data collected with a high flow impactor. So it's kind of analogous to like a Moody or something like that, where you have size-dependent impaction onto offline samples that are then analyzed in the same way as the others that I showed from earlier on.
Yes.
Yeah.
Thank you.
Yep.
This is the first time that's ever happened.
All right, we have a question online.
Yeah, so identification of pollen fragments is kind of tricky. The tools that we were using are these chemical tracer analysis that Betsy Stone at Iowa is leading, where you can essentially look at different sugars that are associated with the different biological aerosols. I can't remember exactly which ones show up in pollen, but we tended to see low concentrations of those pollen-associated sugars. And then the other thing is just looking at ice nucleating particles and potentially their size dependence. But, well, okay, and I guess the other thing too is that we had pollen count data, just looking at intact pollen grains. And so we would expect, it's probably not possible to get huge concentrations of pollen fragments if you didn't have any intact pollen before it started getting humid. So those are kind of the things that we looked at. And the pollen concentrations never got incredibly high during this campaign, despite us specifically targeting grass pollen season.
Any other in-room questions?
Thanks, Russell. I have a question going back to the cloud seeding operations. I don't think you need to go back to the slide or anything. I'm just kind of curious. It's clearly people are doing this because it's effective to some extent. Or I guess my question is, what do you know in terms of how efficient those operations are? Is it pretty reliable, or, and what can you learn from that in terms of the background INP?
I think it's actually a very interesting thing because doing research as to how effective cloud seeding is really hard and expensive, and just paying someone to set up a generator and burn stuff is pretty cheap. And so I don't think it's quite as clear-cut as far as the efficacy of cloud seeding. I mean, clearly people have done experiments where in some cases you can, release ice nucleating particles in a supercooled liquid cloud, you see glaciation, you see precipitation. A lot of those experiments have been done. The one that's harder is like if you have this network of ground sites and you're burning, you're producing silver iodide particles, based on some forecast, how much more snowfall do you actually get? Yeah.
Okay, so it's unknown.
Yeah, it's not necessarily as clear as you'd like.
And I have another question, if it's okay. And then I really liked that you guys had that network that Handix did, and it was interesting to see that the INP number concentrations were very similar, but I was curious if it was also the same across the different sites with surface site density once you accounted for
Yeah, so there's a paper out about the aerosol differences between those distributed sites, and they are often very similar. You're talking about surface site density. Yeah, so I guess the surface site density doesn't change very much because the aerosol is mostly similar across all of the different sites as well. There is some deviation, and that is one that we're trying to look more closely at. Part of the thing that's tough is a lot of those sites only measured to about 2.5 microns, and so they are potentially missing a fair amount of the coarse aerosol surface area. So it's not quite as clear cut.
Great questions. Any others in the room? I don't see any more questions online. So let's thank Russell once again for that interesting talk.